Welcome to Learn Chemistry Together. This video is about protein. Proteins are large biomolecules and macromolecules that comprise one or more long chains of amino acid residues. For example, a representation of the 3D structure of the protein myoglobin showing turquoise alpha helices. This protein was the first to have its structure solved by X-ray crystallography. Toward the right center among the coils, a prosthetic group called a heme group, shown in gray, with a bound oxygen molecule, red. Protein structure. Proteins are generally categorized using 4 levels of structure: primary, secondary, tertiary, and quaternary structure. Primary structure. The primary structure of a protein is the sequence of amino acid residues. For example, primary structure of human insulin. Insulin is comprised of 2 polypeptide chains, called chains A and B, which are linked together by disulfide bridges. Secondary structure. The secondary structure of a protein refers to the 3-dimensional conformations of localized regions of the protein. Depending on the sequence of amino acid residues, localized regions of peptides often adopt particular shapes. 2 particularly stable arrangements are the alpha helix and beta pleated sheet. An alpha helix forms when a portion of the protein twists into a clockwise spiral. Each turn has approximately 4 amino acid residues, and each carbonyl group experiences hydrogen bonding with an NH group that is 4 residues farther along on the chain. In an alpha helix, the R groups, side chains, extend outward, away from the helix. A proline residue cannot be part of an alpha helix because it lacks an NH proton and does not participate in hydrogen bonding. Many proteins contain alpha helices. For example, akeratin, the major component of hair, is comprised almost entirely of alpha helices. Beta pleated sheets are formed when 2 or more protein chains line up side by side. In a beta pleated sheet, hydrogen bonding occurs between the carbonyl group and NH group of neighboring strands. The R groups, side chains, are positioned above and below the plane of the sheet in an alternating pattern. Beta sheets are commonly formed from segments that bear residues with small R groups, such as ALA and GLY. With larger side chains, steric hindrance prevents the strands from getting close enough to participate in hydrogen bonding. Spider webs are comprised primarily of fibroin, a protein containing a pleated sheet arrangement that endows fibroin with structural strength. Fibroin is also the major component of silk. Tertiary structure. The tertiary structure of a protein refers to its 3-dimensional shape. Proteins typically adopt conformations that maximize stability. In aqueous solution, a protein will adopt a conformation in which the polar groups are pointing toward the exterior of the protein and the nonpolar groups are located in the interior. Consider the structure of myoglobin, a protein used to store molecular oxygen. The regions marked in red represent polar side groups, and they are positioned on the surface of the 3-dimensional structure so that they can interact with water, thereby maximizing the stability of the protein. The intramolecular forces holding a typical protein in its folded shape are relatively weak, and under conditions of mild heating, a protein can unfold in a process called denaturation. A common example is the cooking of an egg, during which the protein albumin is denatured and changes from a clear solution to a white solid. The secondary and tertiary structures are lost, but the primary structure remains intact. That is, the sequence of amino acid residues does not change. Loss of tertiary structure is typically accompanied by a loss of function, because the function of a protein is very much dependent on its 3-dimensional shape. The denaturation process is irreversible in most cases. Factors other than heat can also cause denaturation, including a change in pH or a change in solvent. Quaternary structure. A quaternary structure arises when a protein consists of 2 or more folded polypeptide chains, called subunits, that aggregate to form one protein complex. For example, consider the quaternary structure of hemoglobin, which is comprised of 4 different subunits. Each subunit in hemoglobin is displayed in a different color. The subunits have many nonpolar amino acid residues, and as such, there are many nonpolar groups on their surfaces. These nonpolar groups experience van der Waals interactions, which hold the quaternary structure together. Proteins perform a vast array of functions within organisms, including catalyzing metabolic reactions, DNA replication, responding to stimuli, providing structure to cells and organisms, and transporting molecules from one location to another. Proteins differ from one another primarily in their sequence of amino acids, which is dictated by the nucleotide sequence of their genes, and which usually results in protein folding into a specific 3D structure that determines its activity. A protein contains at least one long polypeptide. Short polypeptides, containing less than 20-30 residues, are rarely considered to be proteins and are commonly called peptides. The sequence of amino acid residues in a protein is defined by the sequence of a gene, which is encoded in the genetic code. In general, the genetic code specifies 20 standard amino acids. Shortly after, or even during synthesis, the residues in a protein are often chemically modified by post-translational modification, which alters the physical and chemical properties, folding, stability, activity, and ultimately, the function of the proteins. Some require a non-protein chemical compound or ion for biological activity. These are known as cofactors. Proteins can work together to achieve a particular function, and they often associate to form stable protein complexes. Once formed, proteins only exist for a certain period and are then degraded and recycled by the cell's machinery through the process of protein turnover. A protein's lifespan is measured in terms of its half-life and covers a wide range. They can exist for minutes or years with an average lifespan of 1 to 2 days in mammalian cells. Abnormal or misfolded proteins are degraded more rapidly, often by the proteasome, which is a large protein assembly itself. These proteins are degraded either due to being targeted, ubiquitin ligases can mark a protein for destruction, or due to being unstable or damaged. Like other biological macromolecules such as polysaccharides and nucleic acids, proteins are essential parts of organisms and participate in virtually every process within cells. Many proteins are enzymes that catalyze biochemical reactions and are vital to metabolism. Some proteins have structural or mechanical functions, such as actin and myosin in muscle, and the cytoskeleton scaffolding proteins that maintain cell shape. Other proteins are important in cell signaling, immune responses, cell adhesion, and the cell cycle. In animals, proteins are needed in the diet to provide the essential amino acids that cannot be made. Dietary proteins are digested into amino acids to replenish the body's free amino acid pool, which is mostly used to make new body proteins, but is also critically important for energy production and to make other vital nitrogen-containing molecules. Thank you for watching. Click subscribe for supporting this channel.